The broadcast is now starting. All attendees are in listen-only mode.
Hello, my name is Liz Vickers, and I'm the group publisher for Forensic Magazine. I want to welcome everyone to today's webinar sponsored by Life Technologies titled Ion Torrent Semiconductor Sequencing for Forensic Applications. Today we will learn about Next Generation Sequencing, NGS, that has the potential to vastly expand the capabilities of human identification laboratories, enabling simultaneous analysis of a wide range of forensically relevant genetic markers to aid in resolution of challenging cases. Following today's presentations, we'll have a Q&A segment. I encourage everyone to submit questions at any time during the presentation by typing your question into the designated section on your screen. We will get to as many questions as we can during the time allotted, but if we do not get to your question, we'll be sure to send a reply to you via email after the webinar. The presentation will be available on demand tomorrow, and you will be sent a follow-up email with a link to access the presentation anytime you wish over the next few months. Our first presenter today is Lisa Calandro, the Director of Product Management at Life Technologies. Lisa leads a team of forensic scientists to define user requirements for new human identification products and to provide training and support to forensic analysts worldwide. She received her bachelor's degree in microbiology and immunology and her master's degree in forensic science from UC Berkeley. Our second presenter is Professor Dr. Walther Parson. Walther received his PhD in forensic molecular biology in 1999 and holds an associate professorship since 2001 at the Institute of Legal Medicine, Innsbruck Medical University. He is a member of international forensic working groups, including the ENFSI DNA Working Group, EDNAP Group, and ISFG. We will now begin the presentation with Lisa Calandro, and I want everyone to enjoy today's webinar.
Good morning, afternoon, and evening, everyone. I'm pleased today to be here to share with you some developments on our Ion Torrent PGM system. The first portion of the presentation will provide an overview of PGM technology, some information about the development of a SNP panel for HID applications, and then I'll turn it over to Dr. Parsons to discuss some work that he's been conducting using the PGM for mitochondrial DNA sequencing. So I wanted to talk a little bit about next generation sequencing, which is really enabling a massive amount of data to be generated, megabases to gigabases of data in a matter of hours to days. So this technology is allowing us to realistically consider sequencing as a tool for forensic applications, revealing information that may be difficult to access by other means. Some of the interesting applications that apply to the forensic space include the determination of multiple polymorphisms, including STRs, SNPs, indels, and other types of genetic markers. In the presence of scant or degraded DNA, identity or kinship analysis could be conducted with fragments as short as 60 to 70 nucleotides. So this is an application that really applies to missing persons and disaster victim identification. Other SNP classes, including ancestry-informative SNPs, could be employed in routine forensic analysis for investigative leads and subject exclusion. And haplotyping for kinship, phylogenetics, or analysis of degraded DNA can be carried out by sequencing Y chromosome SNPs or mitochondrial DNA. Sorry, trying to advance the slide here. Sorry about that. The next-gen sequencing system using the Ion Torrent PGM involves three fundamental parts: library preparation, sequencing, and data analysis. The chemistry is performed on a semiconductor chip, and it uses natural nucleotides and natural enzymes. So there is no light-based detection involved in this type of sequencing. So I wanted to talk a little bit about the mechanism of sequencing. It is performed on an ion chip, which contains ion-sensitive, transistor-based sensors in perfect register with 1.2 million wells. And the detection mechanism is effectively by pH. So within the wells, you have a bead, which contains a DNA template which is attached to the bead. So the chip-based technology leverages the cumulative investment in semiconductor manufacturing over the past 40 years or so. So this is an extremely scalable technology. By having the 1.2 or more million wells on a chip, you allow confinement of individual sequencing reactions and parallel simultaneous detection of these sequences. If you look at the cross-sectional view, you'll see a 3-micron microwell holding a bead with a DNA template attached to the bead. Below that well, you have an ion-sensitive layer, which is shown in green, and a transistor stack underneath. So as you flow individual nucleotide bases sequentially through the well, the bases are incorporated if there is a complementary base on the template strand, and the nucleotide releases a hydrogen ion, which creates a pH change in the well. The sensing layer will detect this pH change and translate it to a digital signal. So the voltage detected is proportional to the number of bases incorporated. Another technology that we've developed to simplify the targeted resequencing application is the ion AmpliSeq technology. So this is a custom design service with a bioinformatic pipeline and a system-specific chemistry that allows extremely high levels of multiplexing. So it's very similar in terms of the workflow to a straightforward PCR, that you can multiplex several thousand targets. You can sequence as little as 10 nanograms of DNA from a variety of sources, including very challenging substrates such as formalin-fixed, paraffin-embedded material. And you don't need to purchase any expensive microfluidic PCR platforms in order to pool and normalize the amplicons. And you can basically go from DNA to annotated variants in as little as 10 hours. There are a number of different chip sizes that you can select from depending on the amount of sequence you need to generate. And the capacity and coverage of a particular chip is determined by the number of wells and is a function of the sample size, which is the target length multiplied by the depth of coverage. So for example, if you have a 16 kb target and you're looking for a minimum coverage of 100 reads, you would be sequencing about 1.6 megabases, which would allow you to either do 30 samples on a 314 chip or 96 samples on a 316 chip. If you want higher coverage, which you might want for somatic applications where you're searching for rare variants, you would not be able to sequence as many samples on a chip, so you would do 3 samples on a 314 chip or 15 on a 316 chip. The workflow starts with preparation of a library, followed by clonal amplification onto beads. You can select for beads that have sequence attached, and then load those beads onto the chip and perform the sequencing and data analysis. So I'll go into a little bit more detail on that. So this is an overview of the workflow. You start by preparing the genomic library by fragmenting the DNA, and there are different ways that you could do this. You could either generate specific amplicon sizes using AmpliSeq, or you can actually shear the DNA. Once you fragment it, you attach your forward and reverse adapters, and you clonally amplify these onto beads. So you are basically attaching your sequence to the beads. You use a magnetic bead-based enrichment process to select beads carrying template. And then you also have your sequencing primers and your DNA polymerase bound to the template carrying beads as well. You pipette those into your chip's loading port, install it in the instrument, and then your sequencing is performed by cyclically flowing your nucleotides across the chip over an approximately 2-hour run. And then you utilize signal processing software to convert the raw data into measurements of incorporation in each well for each successive nucleotide flow. So after the bases are called, each read is passed through a filter to screen out any low accuracy reads, and you generate per base quality values for each incorporation. So this is a schematic of the sequencing flows and cycles. So a flow is basically the event of exposing the chip to a particular DNTP, so T, A, C, or G, followed by a washing step. So each cycle is a consecutive flow of the individual nucleotides. So as you flow across the T, it will bind to its complement. If there is no complement, it is washed away. And so you're sequentially adding to that sequence and releasing hydrogen ions as these bases are incorporated. So the next cycle is basically the same thing, flowing the bases sequentially, and so on until you have filled in the sequence. So in the case where you have homopolymers, you'll add bases to each of those locations sequentially. The data output is in the form of an ionogram, and you need to read this ionogram both from left to right as well as up and down. So the height of the bar indicates how many nucleotides are incorporated during the flow. So where you have homopolymeric sequences, as you can see here with 2 A's and 3 T's, you get higher bars indicating the number of bases repeated. For this system, we can typically detect 6 to 7 bases for homopolymeric regions, but as the chemistry and bioinformatics continue to improve, our ability to read through homopolymers will also improve. So I want to talk a little bit about the development of the HID SNP panel. We released an initial panel back in July, and this was an identity SNP panel. And the objective here was to combine a set of SNPs for the purposes of identification with high heterozygosity and low population heterogeneity. We are preparing now to launch our version 2.2 identity panel, and this is based on published SNPs, and it's a combination of the 52 SNP for ID SNPs that were used in the GenFlex program and also used quite frequently throughout Europe for the Euro 4Gen project. The 92 KID SNPs and 35 Y SNPs, which were improved over the original panel to allow discrimination between high-level phylogenetic clades. Within the 92 KID SNPs, there are 45 unlinked SNPs that allows us to achieve these very high genotype match probabilities. When we look at the different methods for library preparation, the long amplicon method is targeting longer amplicons and then bringing those amplicons down to the appropriate size for PGM sequencing using a shearing process. For SNPs, we're primarily using the short amplicon method using the AmpliSeq design process. So we don't need to do any shearing. The process of preparing the library includes the ligation of adapters. P1 adapters allow attachment to the bead. The IA adapter initiates the sequencing, so this is where the sequencing primer will bind. And then you have the option to also incorporate barcodes. So barcodes are 10 mers, and we have 96 unique barcodes available. And this allows you to potentially multiplex samples in the same sequencing run. So it's basically an identifying sequence that allows you to determine which sample the sequence is originating from. Much of the power of the system is in the data analysis. the sheer volume of sequencing information generated necessitates a highly capable bioinformatics system. So we have available a Torrent server and a Torrent browser that allows a simple data management in an easy user interface to avoid the need for a bioinformatics manager to handle all of the data. So you can display all of your runs in the browser, and you can easily access those runs. From the browser, you can create a run analysis report. This provides an overview of the mechanics of the run, how the chip worked and some of the data quality values. And then you can also launch plugins. And we have application-specific plugins that allow you to look at particular types of data. Of course, you can also use a variety of third-party software desktop tools that allow you to further interrogate the data for the application of your choice. So this is an example of an amplicon coverage plot, and this allows us to assess the success of our panel. And the success of the panel is a function of the level of the coverage and the evenness of the coverage. So what you see here is the SNP panel, the original SNP panel with data from a female on the left and a male on the right. So you can see where the Y SNPs are located. We don't have any data there for the female sample. What the coverage plot is showing you is the number of reads that were obtained for each of the SNPs, sorted from left to right by depth of coverage. The yellow is the forward strand, and the green is the reverse strand. So you can see a couple of instances here of strand bias, where one strand we were able to sequence and the other strand didn't perform quite as well. So we're really looking for SNPs where we have good coverage and good even reads between the two different strands. And typically we're looking for a minimum of about 20x coverage to achieve confident calls for each SNP. We've also created a plugin for our SNP panel, and this plugin allows you to look at each individual SNP location. So if you look at the top here, it gives you the level of coverage. So we had 544 reads, of which 538 gave us an A at the SNP location. And it also allows you to assess the forward and reverse strand coverage. And finally, the frequency of calls for the particular base. So this would be an example of a homozygous individual at that particular SNP. If you look down to the third line, you can see an example of a heterozygous result where you have approximately equal amounts of reads between A and G. So this kind of plugin gives you a lot of information about your individual SNP locations. You can also look at the data with an allele coverage plot. So on the top is the number of reads for each particular base call. And on the bottom, you can see that same data normalized. So you're looking here for occurrences of potential heterozygotes, where you see about 50-50 coverage for the two different bases versus homozygous coverage, where it's primarily a single color. So next generation sequencing technology has allowed us to achieve high levels of multiplexing, both for SNPs and the combination of SNPs and other applications, as well as individuals. We are preparing to release our Identity SNP panel version 2.2, which uses well-characterized SNP polymorphisms, and will be available to collaborators who are really looking to move to next-generation sequencing as a SNP detection system. And along with that, we have a SNP plugin utility, which was built to assist with the display and the data evaluation. And this is something that could also be used with custom SNP panels that you might wish to develop yourself using the AmpliSeq pipeline and the tools that we have available. So where are we going from here? We're also looking to develop ancestral informative and phenotypic SNP panels. So the application here would be to exclude suspects and provide investigative leads in criminal cases where potentially a suspect has not been identified. So we're looking for SNPs that allow a high degree of discrimination between ancestral groups and allow for determination of certain phenotypes such as hair and eye color. So our initial panel design is based on some published information and access to some data that we've received from various collaborators to help us design the appropriate SNP panel for these applications. We're also looking at STR analysis on the PGM, and we have had success in terms of determining genotypes by sequence length, but we also are able to detect additional differences between individuals even if they have the same allele repeat structure. So this is one of the values of sequencing. So for example, we have an individual, two different individuals, both with 15 repeats, but we were able to detect slight differences in their actual sequence, which would allow us to differentiate those individuals in an actual forensic case scenario. Before I pass this over to Walter to talk about his mitochondrial work, I do want to acknowledge our R&D team here in Foster City and our collaborators and advisors who have guided us in the development of these panels, including Dr. Ken Kidd, Dr. Art Eisenberg, Dr. Bruce Spadoli, and of course, Dr. Parson. So with that, Walter, I will turn it over to you.
Great. Thank you so much, Lisa. And now you're going to hear from Dr. Walther Parson.
Hello, everybody. I see that there is a lot of you out there. Of course, I don't see who you are, but this is why I'm holding a rather general presentation on the work that we did. And what I would like to tell you today is our experiences with mitochondrial genome sequencing on the PGM. The reason why we have started to look at PGM with mitochondrial DNA is because we have a research interest in that molecule, and we also do have a database called MPOP, which is a internet database that can be used by forensics to query frequencies of haplotypes. This database contains control region sequences so far, but we have some research work going on to go for full mitochondrial genomes there as well. So as for the mitochondrial DNA, as I mentioned, the control region is the only larger non-coding part which is currently used in forensics. However, there's much more information out there in the remaining portion in the coding region, which would be very suitable as an additional discrimination power for forensic purposes. The reason why we like mitochondrial DNA in forensics is because it's present in a much higher copy number per cell than nuclear DNA. And that is especially true for samples that are known as difficult or challenging samples that contain very few or no nuclear DNA amounts. For example, hair shafts are such samples that are commonly found in the forensic field and that usually do not give results with STR typing, but are showing quite high success rates with mitochondrial DNA. As you know, mitochondrial DNA is inherited maternally. There is no recombination yet found in this molecule, which means that with an analysis of the mitochondrial DNA, we are identifying an entire maternal lineage rather.
Walther, I'm just going to Walter, I'm going to interrupt you for a second. For some reason, your screen isn't showing. Did you click on the get the screen? I had passed it to you. We're not seeing your screen.
Let me see what I can do here.
Tim, if you're on there, maybe you can see if you can pass it. It's like he all of a sudden dropped off there. Up there it is.
I think it should be better now.
Yep, it's fine. Great. Sorry about that, everybody.
Oh,
we'll continue.
I'm very sorry. So probably you didn't see the slides. This was my introduction slide here. making reference to the MPOP database. That's something that most of you know. Here is the slide with the elevated mitochondrial DNA copy number that I was referring to, and hair shafts, some of which do contain nuclear DNA, but the majority do not, and those are the ones that we want to analyze with mitochondrial DNA. Here is the slide for the maternal inheritance, and here we are back again in the streamline of the presentation. And this is the application where we use mitochondrial DNA both for forensic analysis but also for anthropological work. When we have old remains of deceased people present, then there are actually two advantages of applying mitochondrial DNA. We have the high copy number that increases the chance of getting a useful result, and we do have the maternal inheritance that allows us to test for relatedness across several generations or between individuals that are deceased. Due to its mode of inheritance, mitochondrial DNA is inherited along a phylogeny. The same is true for the Y chromosomal part that has been used by population geneticists to reconstruct human migration, and that also leaves useful information when we look at the mitochondrial DNA in a forensic perspective. So why would we be looking at mitochondrial genomes? Well, if we were able to type the entire mitochondrial DNA, of course we have an even much higher discrimination power that we currently have with control region typing. Another advantage is, of course, that we are looking at phylogenetic signals, which have two major advantages for forensic work. First of all, it tells us something about mutations that happened during human evolution and gives us the possibility to calculate actual mutation rates, which we may want to introduce when we interpret forensic evidence. And on the other hand, the phylogenetic signal is very useful to determine the quality of typed data in the laboratory. We are regularly performing QC checks of data from the literature or data that are submitted to publications, and those quality checks that we perform are majorly basing on the phylogenetic signal that is out there. I want to mention that we do have a running NIJ project together with AFDL where we are looking at the potential of full mtGenomes for forensic purposes. Why haven't we used the PGM? Our experience with next generation sequencing has been very scarce, so we were relying on data that we saw from different laboratories, experiences from different groups. We saw that the system seems to be very sensitive. It is amenable to degraded DNA. It has a fast library construction process and an automated enrichment system, which I think is useful when we think about a laboratory process in the forensic setting. Also, it does have a very fast sequencing time and a high throughput in terms of runs per week. And again, that could be very beneficial if we think about the application of forensic cases in terms of short reporting times to the court and to the police, things like that. The samples that we have been using for the tests and the exercises that I would like to show you are samples that come from different mitochondrial phylogenies. So we were selecting samples from South Africa, from the South Asian part of the world, and from West Eurasia, mainly Europe. We were also looking at different sources. So we were testing blood as a source, buccal swabs, and paraffin embedded tissue, knowing that we do have different qualities of DNA. We do get different degradation states of DNA and want to see how this performs with the tested methods. Now we do have a Sanger-type sequencing protocol here in our laboratory, which we use if we want to type full genomes. This involves the PCR amplification of the entire genome by either 2 or 9 overlapping amplicons that are then Sanger sequenced using a little more than 100 primers. This usually results in very nice sequences. I'm not talking about difficult case, for example, at this point. I'm talking about samples that contain a fair amount of mitochondrial DNA and allow one of these two strategies to be typed. However, this method is laborious. The throughput is very low, as you can imagine, and also the analysis time that is required to produce such a consensus sequence is quite high. With the PGM, we were trying to start from exactly the same point as Sanger sequencing in order to be able to compare the different techniques. So we were producing a library with the PCR of 8.5 kilobase per fragments. After PCR, they were enzymatically sheared to reach the fragment size that is required for the chemistry of the individual chip. Then the library was prepared with the adapters, clonal amplification, and the positioning of the amplicons on the ion spheres, just as Lisa has explained. We have been using 316 chips, and we have been using two types of chemistry, a 100 base pair chemistry and a 200 base pair chemistry. And then the data analysis follows the same stream as we just heard before. What kind of data do we get when we use PGM? Well, first of all, two important files that are an output of that technology is the binary alignment map and the binary alignment index. These are two files that can be used for the analysis on the variant caller that we just saw before, but that is also a file format that you can use to look at third-party software in trying to look at the individual alignments. And that's what we did. We analyzed all the data with the Torrent browser variant caller. We were using this version. This software is using a TMAP Smith-Waterman alignments. It does not show the actual alignment as a view. This is why we were looking at different software like IGV. This is a freeware that uses a paired read alignment, so it's different to the one that we just saw above here. But it also shows a graphical view of the alignment of the individual sequences. We also tested NextGene that also uses a paired read alignment and does a visualization, and Sequencer that has a tablet for next generation sequencing integrated, and Sequencer is using a GSNAP alignment. So we are looking at different softwares and different alignments and see how they involve or how they affect the analysis with next generation sequencing on the PGM. After a run, you get a couple of information about the run, some of which are very useful. For example, the loading density on the chip. As you can imagine, you work with this, but you never get a feedback how well you are performing. Here you see the coverage of the individual amplicons targeted on the chip, and the more red it's getting, the more coverage and more density is there. So this is a good sign of a very useful and a very successful run. And further down the road of the analysis, we are coming to the point when we get information about the individual variant cause. Lisa was guiding us through those. I'd like to repeat this for mitochondrial DNA real quick. Here is the reference sequence. We are aligning to a revised Cambridge reference sequence in mitochondrial DNA forensics. These are the positions according to this molecule. We do see the reference base at this position. We see the variant base. We see the frequency with which that variant was observed under this total coverage, and here is the variant coverage. So we know at position 13708, which is a G in the reference sequence, the variant was an A, so it's a transition. It was found 406 times under 410 coverage, which results in a variant frequency of 99%. Now we have been using, as you saw before, quite diverse nature of samples, and this is why we applied quite a high threshold in terms of the variant frequency that we would report for the comparison with Sanger typing. And that's only because we were using different qualities of DNA. For high quality, this threshold could be set much lower. But just for you to understand, the following comparison is done with a 20% variant frequency threshold. Here is a quick overview how the other alignment softwares look like. This is IGV, and the table now it's presented here. This is a snapshot of NextGene and the way how the sequences are aligned here. And this is a picture of how this is done in the tablet with Sequencer. This is probably the most important slide in my presentation because it summarizes all our findings. We did have 100 base pair and 200 base pair sequencing chemistry. We have been looking at 31 mitochondrial genomes with the 100 and 33 with the 200 base pair chemistry. This is the number of bases that were actually read and compared to the Sanger type sequence. We did find 95 differences in this number of base pairs between PGM and Sanger for this chemistry here, 100 base pair chemistry, and we did find 81 differences in this number of base pairs for the 200 base pair chemistry. So this is total number of differences for two settings. If length heteroplasmy was present, we didn't consider this. So if there is a difference in length heteroplasmy between PGM and Sanger sequencing, we didn't take this as a difference at this point because this requires a different experimental setup. And also, as you know, length heteroplasmy is disregarded in forensic analysis anyway. Also, we see that the variant caller is using a 5' alignment of insertion deletions, which means that they are put to the 5' end, whereas in forensics, we are used to look at the 3' end alignment. So if we saw differences here, we also didn't count this as differences between technologies because it's a convention. So after all, we were very surprised by this very low number of differences that we find here. You know, from the literature, I was expecting something like a 1% error rate is what they usually report, and you never know what they mean with error rate. But here, if we call difference an error, we would be 50 times better off regarding this issue. In the following, I would like to show you where the differences happened. And if you look at these pie charts, you see that 2/3 of the differences are observed in the HVS-1, HVS-2, and HVS-3 C tracts. These explain about 60% of our differences. We do see additional deletions outside the homopolymeric tracts, and we do see substitutions differences outside the tracts. And apparently there is a little difference between 100 and 200 base pair chemistry, which I come back in a minute. So let's go in more detail here. If we look at the HVS-2 tract, the reference sequence has 5 Cs between this T and this G at positions 310 and 316. However, most of the sequences, more than 99%, do have 6 Cs. And interestingly, when using the TMAP aligner, we got differences in all samples with respect to 315.1. So these were not reported by the variant caller, PGM, however they were present with Sanger type sequencing. Well, we don't think that this is an artifact of the PGM. We think this is an artifact of the TMAP aligner because we did the following experiment. We modified the RCRS and introduced an individual C so that it has 6 Cs. And what we saw as a result is that the variant caller brought confirmatory results to the Sanger type sequences. That means when the TMAP aligner is using a reference sequence that differs by deletions, then there is a likely chance that this is not reported in the output. However, if it's a de novo assembly or a reference that does not contain insertion deletions, then this is not affected. So apparently, the data are there, the PGM is sequencing and reading that, however the alignment may have an effect on that. And if we use a paired read aligner, as I mentioned before, then we don't see the lack of 315.1 Cs in the sequences. Now, similar situations appear in the other C tracts, and I'd like to go really fast through them. There is another portion of 7 Cs between this A and T here. The reference sequence variant has 7 Cs. They were all reported in agreement between Sanger and PGM. However, if there were insertions, we did not see it in the output of the T-map alignment. However, if we use different aligners, as you will see in an example later, you see that even the insertions here can be sequenced by PGM. So more than 7 Cs are possible to be read here. In the HVS-1 C tract, we have a situation where 16189, a T here, is separating two small stretches of 5 and 4 Cs. This position is prone to a transition, and if this happens and we have a C here, then we get an uninterrupted stretch of more than 10 Cs, which usually results in extensive length heteroplasmy. In our sample set, 3 samples of the 100 base pair chemistry and 5 samples of the 200 base pair chemistry were affected, and those were not reported with the variant caller. However, if we take a look at the paired read aligners, we do see that some of this information can be captured. We are looking at genome 28, and this is the haplotype only with reference to the C stretches. This is how NextGene is working with this information. NextGene is reporting a 309.1 C, although with a relatively low score, and it's a different nomenclature that they use. And it also reports the 315.1 C, again with a different nomenclature. It is reporting the 16189 C that is present in this sample, and it is not reporting in the table a 16193.1 C, but if you take a look at the alignment down here, you see that some of the sequences do have the insertion between 16193 and 16194. So if we take this entire stretch here, we are getting a confirmatory read between Sanger and PGM with NextGene of about 11 Cs, which was very surprising to me because I thought that next generation sequencing technologies are not able to read more than 8 or 9 Cs. In terms of the substitutions, we found 29 in the 100 and 15 in the 200 base pair chemistry. The vast majority, 3/4 of the substitutions, go back to 6 samples. The problem was the DNA was just too low. We got too low coverage values. We didn't decide to sequence them with the 200 base pair chemistry because they even gave us a hard time using Sanger type sequencing for those. So if we subtract those 22 here, we end up with 7 for the 100 base pair chemistry and 15 for the 200 base pair chemistry. There is an interesting observation because some of those 15 are found in multiple samples. Like these 3 positions here were found to differ between Sanger and PGM in 3 samples. Now, those 3 samples, those 3 positions are very close to our PCR primer binding site with which we are producing the library. We do not fully understand the reason why we see differences here, but there's apparently a system going on here. And again, this is something that we observe with the T-map alignment and not necessarily with the paired read alignment. Here is another position that showed differences. It's a 405.6T, which is a signature mutation for haplogroup H5, of which we had 4 samples in our sample set. It is a substitution, but we are looking at an insertion deletion event because there is a short T stretch 5' of this region. And if there is a 456T, then this stretch is prolonged by a single T. And again, I think it is, since we only saw this with the T-map alignment, we believe, we have confirmation through paired read alignment, that it is read by the PGM, but it's very much depending on how the alignment and the caller is dealing with this information. This finally brings me to the deletions. I'm talking about deletions outside the hypervariable tracts now. We observed 11 in the 100 base pair chemistry and 13 for the 200 base pair chemistry. Here is an interesting observation. A total of 13 for both chemistries were reported as a deletion together with a substitution at the same position. Usually they are accompanied by quite low coverage values. The substitution itself was confirmed by Sanger sequencing, but the deletion was not. And apparently, again, we think that this is may be related to the TMAP aligner because we do not see such position with other alignment in softwares. At this point, I need to mention that in general, the TMAP aligner is performing very well, and if we were using other aligners, they also do have other problems. But since the study was set up in a way that we were using the variant caller as the reference method here, I'm reporting the differences and then the exceptions to these differences with different software. Finally, I would like to talk about a deletion event outside the stretches that was not reported by the PGM for one sample. Well, I should say by the variant caller for one sample. If we take a look at the IGV viewer, we do see that it is reported, however, with a 5' alignment here. And again, it's a deletion in a little homopolymeric region. And very likely, it is again the TMAP aligner that is producing or that is not reporting this deletion because the reference sequence doesn't have this deletion. Okay, that brings me to the summary. We do have a substantially higher throughput with PGM compared to Sanger. That's what we expected. This is the hypothesis of the approach. We were very surprised about the high concordance already at this stage between Sanger and PGM. Again, we were expecting 1% differences. We do see 0.0 below 0.002, and I think we were very conservative in terms of treating differences. So we were also looking at the Ctracts in terms of differences. This is all done again under a 20% variance threshold. As I said, 2/3 of those are in the Ctracts, and the alignment has a great impact in whether they are reported or not. Some of the deletions outside the Ctract have been observed together with substitutions. Also, I do think this is something that has to do with the alignment algorithm. As we speak, the software is constantly improving. We did get updates of softwares that improve the situation very much, and I see that there is a lot of research and development going on. We do see that the TMAP aligner is performing very well if we look at de novo sequences. If we compare to a reference, then we do see that other aligners work better, like the paired read aligners, if it comes to the reporting of insertions or deletions relative to a reference. All right, I'd like to thank a couple of funding bodies that provided the relevant resources to carry out the work. I'd like to thank ABI Life Technologies for many hours of support and collaboration. I would like to thank Christina and Gabriela from our laboratory. They are experts on the PGM. They did a lot of work, a lot of fantastic work with this instrumentation, and also Bettina and Liane for providing samples and doing Sanger-type sequencing work. And finally, I'd like to thank our collaborators that provided samples from Portugal and from South Africa. And with the final slide, I would like to turn back to Lisa, making the reference to the ION community webpage, through which the communication with the instrument and with the scientists is enabled.
Great, thank you, Walter, and thank you for the reminder that it's Valentine's Day and we need to run out and get cards. So for those that are interested in the PGM system and other offerings that we have around the ION Torrent technology, please do visit our ION community webpage. Under the applications heading, you'll find HID applications, and we have specific protocols posted there. It's also a great site to interact with other scientists that are using the system and get your questions answered and share your experiences on the system. So thank you very much for attending, and we're happy to take some questions.
Okay, great. Thank you so much, Lisa and Walter. We're now going to move on to our Q&A portion, and you can enter questions at any time, so please submit them in the space provided. We have some great questions that came through already, so I'm going to begin. Regarding the ION chip, when you add your sample to the chip, can this be a mixed sample, or does it have to come from one person?
So there is the potential to add a barcode adapter to your samples, and those are added to individual samples, and then it becomes part of the sequence. for each of the targets coming from that individual. So you can pool individual samples on the same chip, and then you will have basically an address for that sample in the form of the barcode. So definitely that is one of the major capabilities of the system, that you can multiplex individuals and individual samples.
Great. And the second part of that question, they were also wondering how much the Ion Torrent PGM costs.
So pricing is variable depending on the region, but it is a relatively inexpensive offering for sequencing. I believe that with the entire system, it's less than about 100,000 US dollars.
Okay, and the next question is the Torrent server housed at the user site or at Life Technologies?
The server is part of the system, and it's housed at the user site.
And what happens with the lower input amount of DNA, example 1 nanogram or less? Can you still get good sequencing results?
It is possible to get sequencing results with 1 nanogram or less. Possibly Walter can speak to some of his experience with the mito sequencing. I know that he had some samples that were quite challenging.
Well, if you talk about nanograms or picograms, we need to differentiate between nuclear and mitochondrial because those equal different copy numbers. I think it is possible, and our data suggest it is possible to get reads from much lower amounts than 10 nanograms. It depends on the coverage finally. It depends on the overall coverage that you get, and it depends on the coverage of the variant. So it's difficult to give a general answer, yes or no. I think there is some parallelism to what we are used in the forensic community when we look at STR multiplexes. If we do have very low DNA amounts available, we start getting dropout of loci, or we are getting partial profiles. And this is very similar to what we are seeing here with the sequencing with the PGM. But yes, the answer is it's definitely more sensitive than the 10 nanograms that we see listed.
Okay, thank you. How can we get access to the HID genotyper plugin?
So the genotyper plugin is available on the ION community webpage.
And there will be links to that too at the end of the presentation as well. Does the forward and reverse sequencing reaction occur in the same well?
Yes.
Okay. And Walter, this is a question for you. Can you detect DEL 3107?
The answer is yes, you can. However, it is not reported because it's an artifact from the from the earlier version, I should say, of the reference sequence. Some softwares do report it, and others not. One should know, or for those who are not aware with this fact, the Cambridge reference sequence was resequenced and published in 1999, 18 years later after the first attempt. They found between 11 or 14, I can't remember out of my head, differences between the two sequences which they refer to as errors. And one of them is that there was a deletion with respect to the earlier version that the scientific community decided to keep. Because if we would not keep this deletion, then the numbering of all sequences further of position 3106 or 07 would then be changed, and this would lead to a great confusion. So this is something that needs to be dealt with the alignment software and with the notation of the sequence. But it's, I mean, it's very obvious if you think through it.
And is the instrument currently being used for clinical use, for example, genetic disease detection?
Lisa, are you going to say something?
I'm sorry, could you repeat the question, Liz?
Oh, sure. Is the instrument currently being used for clinical use, for example, genetic disease detection?
So that's definitely an area that we're looking into in terms of developing collaborations in other parts of our business. I personally don't have visibility into that, but definitely that is an application on this system. Absolutely.
Great. And when will sorry, I just lost the question as other ones were coming in. When will the HID 2.2 kit be available?
Next week or so.
Yeah, we're hoping to have that available to send out as early as next week.
Great. How close to completion are your studies for ancestral hair and eye color and STR?
We're still in the process of evaluating the various SNPs in terms of their performance. So we're working on vetting out the panel, but we think within the next month or so we'll have something available.
Okay, great. And is access to the ION Torrent server restricted to the lab using PGM via IP address?
It's really up to the lab how they set that up within their IT structure.
Okay. And do you see the PGM instrument replacing the current CE instruments?
You know, right now we're looking at some very unique applications that we feel that the PGM is ideally suited for, so particularly very challenging samples. It does give you some capabilities around multiplexing different targets, but you know, we have a long history in the forensic business, and I think it'll take some time really for people to evaluate the technology, become familiar with it. So I don't see it happening in the very near future, but I definitely think that there's a lot of interest, and there are many forensic scientists around the world who are looking at the system for various applications, some of which are very unique.
And this is a question to Walther. When do you suggest mtGenome sequencing would be available in real casework?
I think there are two answers to that. First of all, there is a technical answer. Remember, we have been amplifying whole genomes with quite long amplicons. That needs to change, and we are currently working on that. But I think it is doable, and the technical solution will be there much faster than the second issue, which is the political solution. Some countries do have reservations in the legislation system in allowing coding region area to be investigated in a forensic case. So I don't know, obviously don't have an answer to when this will happen, but as a scientist I concentrate more on the first part, and I hope that we will be able to see that coming in the next months.
And can we carry out autosomal and YX, STR, and SNP sequencing along with mitochondrial DNA simultaneously on the chip?
Yes, that is theoretically possible, and we're working on refining all of those applications and then ultimately potentially combining them into a single tool.
And here's another one directed to Walther. Do you envision the EMPOP will evolve to include full mitochondrial genome data in the foreseeable future?
The answer is yes. The project that I have been mentioning, the NIJ project together with Ethel, we are working already on full genomes that we sequence with Sanger for several reasons. One, to increase the body of forensically sound data for databasing, and second, with Sanger type sequencing of the full genome, we get a better understanding of the problematic areas, as we did with the control region. We now have a very good understanding of what can go wrong in the control region, and we use this information in order to quality control other data sets. And this is also happening right now with the full genomes. So the MPOP database with the next version will hold full mitochondrial genomes.
I think we have time for like one or two more, and any we don't get to, I'll make sure to forward over to today's presenters, and they'll get back to you via email. I understand how you multiplex using barcodes, but a mixture sample would have the same barcode. Can you use the frequency of SNPs to determine if there is a mixture and what proportion the mixture is in?
Yeah, so that's correct. If you barcode a sample, you'll barcode all of the DNA in that sample. So if the sample is a mixture, all of the targets will have the same barcode. But basically, you're assessing the number of reads from individual sequences. So it is possible to do a mixture analysis by using the number of reads as your quantitative value.
Okay, and this will be the last question. How long does the entire process take from start to finish, specifically mito sequencing?
Is that a question for me?
Sure, you can take it.
Sorry, please say again.
Okay, let me just get back to it. How long does the entire process take from start to finish, specifically mito sequencing?
I think it's the entire process is taking a few days, certainly less than a week. I would say 2 to 3 days. But I never did the work in the laboratory, so I am the one receiving the sequences, and I should ask Gabby and Tina for a real clear answer here.
Well, we'll get back to you if there's any other questions there as well. But I want to thank everyone for attending the webinar today, and thank you so much to our great speakers, Lisa Calandro and Dr. Walther Parson, for the informative discussion. Our contact information is listed here. Also, feel free to email us with any further questions, and we will get back to those that weren't answered. Also visit www.lifetechnologies.com and these other links listed for more information. You will get an email tomorrow with a link to the on-demand version, so you can view this again within the next few months. And I want to thank everyone for attending today's webinar and wish everyone a great Valentine's Day.
Bye-bye.
Bye. Thank you.